Um, and here are some specifics about American coffee that everybody who went to Starbucks sometime can feel about these inventions. And often we continue this discussion in the evening when we had dinner together with family. And I think what comes now from Bernard, I think, is also very important. I have to say I have been very lucky to have very well-trained and creative young scientists, and the majority actually came from Italy. Students like you who had learned very well, who were open and motivated to come. And when Marco, after being very successful, came back to Florence, both Bernard, and he mentioned it here in his quote, and I were very disappointed that he didn't get an appropriate position. He came back, he had trouble getting a position at all, I have to say. And he was quite upset about that in some conversations that we had. And probably in England or in Germany, he would have gotten a career development grant. Currently, these are €1 million or 1.5 million euros. They give young researchers 5 years of time to establish themselves. And Marco was really tremendously gifted, and as you saw, had impact. And this was really a great pity that he couldn't come back and flourish as he was supposed to do. And finally, one quote from Giovanna. She didn't know Marco as closely as we did, but she said, in preparing this event for Marco, I've come in touch with many of his friends and colleagues who have all turned out to be great people and who have all been very eager to work in his memory to celebrate him as we do today. And with that, I would like to finish and thank you for your attention. In the moment, rather slim.
Okay, so we have another question from the University of Chieti, which is, what is your opinion on the role of vitamin D?
Yeah, so vitamin D is interestingly now identified as an important environmental risk factor, not only in MS, but also in a number of other autoimmune diseases. And there are different theories how the vitamin D may play a role. Calcitriol, the active vitamin D, has many effects on the immune system, regulatory and others. One theory that George Ebers' group formulated about the role of vitamin D in the context of MS is that low vitamin D levels lead to differential expression of certain HLA molecules during ontogeny of an individual in early life and shape a T-cell repertoire or an immune repertoire that is different than at high vitamin D concentration. So one influence is that vitamin D itself has immunoregulatory functions, and lower levels may be conducive of having less regulation. This is a simplification, I'm aware. And the other one is that it has an influence very early during life in shaping the immune repertoire by regulating gene expression of HLA molecules and probably also other molecules. The latter theory, I think, still lacks experimental evidence. It's an interesting one. It may be true, but there's still data, and it's, again, very difficult to study.
Okay, so we have another question from Como. Could APLs also modulate the pattern of differentiation of activated T cells, e.g. polarized to more Th17 or Treg, or something else? Does any evidence exist in this regard?
Yeah, I mean, an APL peptide, as many groups, including ours, showed, they can have different functional outcomes. They can partially activate a cell. So if you imagine a Th17 cell that makes IL-17A, IL-17F, other cytokines, a partial agonist or an altered peptide ligand could only activate some of these functions, proliferation in the absence of cytokine release. That is entirely possible, and for Th1 and Th2 cells has been shown very well. It could antagonize, so it could completely shut down the response of the cell, probably by interfering with signaling inside the T-cell receptor complex. And then if an immune response develops de novo, an altered peptide ligand can lead to a cytokine skew towards another cytokine release phenotype than the Th17 type. As I said, T cells are probably much more plastic in terms of their functional profile than we believe. They are not very early committed to doing only one thing or a few things, but can be shaped by whatever signals they encounter. So in principle, I can envision that a Th17 cell still can be skewed unless it's really terminally differentiated. Then I would not expect that just modifying the peptide ligand would lead to a different outcome in terms of cytokine release. That I do not believe, and I'm not aware that anybody has shown this.
Okay, thank you. So another question from Milan, from the San Raffaele Institute. Is such a T-cell-centric vision of MS pathogenesis actual and updated?
Of course it is T-cell-centric because I'm T-cell-centric, but on the other hand, if I had gone into now the role of CD8 cells, of dendritic cells, of monocytes, of type 2 macrophages, NK cells, NKT cells, and I could go on with this list, that have all neutrophils, eosinophils, basophils, have been shown to have some role in some context, in some patients, in some aspects of the disease evolution, and then also CNS cells, we would still be here in the next, at 6 o'clock tonight. I do believe that as an abstraction, that CD4 T lymphocytes, based on what I showed you, between 10 and 50% of the genetic risk of MS comes from HLA class II. And the only cell that really uses HLA class II to recognize something and to exert its function are CD4 T cells. And not only that, CD4 T cells are central to orchestrating many things in adaptive and innate immune responses in terms of functional development and others, maintaining cells alive or propagating them. So I think as an abstraction, it is fair to be T-cell centric, but that of course is not the whole story. As an effector cell, CD8 T cells may be much more important, or innate immune cells. Antibodies may be more important as effector molecules than CD4 T cells. But in terms of induction and maintaining the disease, I'm fairly convinced that CD4 T cells are the single most important factor in our immune system.
So another question, always from Milan, but from the Besta Institute. So with regard to APL, would you expect them to have a role in the generation or stimulation of antigen-specific Treg cells, T regulatory cells?
Yeah, I think this APL phenomenon, and actually Tonegawa's group, when they showed that variant peptides can be important in the selection of our thymic repertoire, I think it is, I have no doubt that an APL peptide could be engineered that also expands Tregs. But it has not been tried, I have to say. So far, at least I'm not aware of it, that somebody specifically created a variant peptide that expands T regulatory cells or leads to their differentiation. And maybe our study, our negative study that led to disease induction, was one of the reasons why APL peptides as a therapeutic agent have been, I don't want to say fallen into disbelief or are neglected now. But this field that was very active and optimistic in the mid-90s to 2000 then more or less shrank down. And I think APL peptides would be particularly interesting in the context of tumor immunology, because as you saw, we could elicit a very strong response by a variant peptide against which natural tolerance is probably not functioning very well. And in the tumor context, I think this would be a very interesting concept that we have been starting to work on.
So now we have a question from the Imperial College in London, from Paolo Muraro's group. So the question is, has Marco or others looked at CD8 MHC class 1 restricted T-cell responses to myelin antigens, to myelin targets?
There have been some studies, for example, by Bill Biddison at the branch where we used to be, who was the head of the molecular immunology section there. In the case of class 1 restricted T cells that recognize myelin antigens, the path, this was at the time when the peptide binding motifs for HLA-A2 and others were just determined and binding prediction algorithms were invented. And what Bill did is he used these binding prediction algorithms to predict peptides in PRP and MBP, and then made tetramers or selected clones by using the peptide immediately. Overall, the body of data for CD8 cells is much smaller from various reasons. They are more difficult to grow, at least autoreactive CD8 cells. And obviously, they should get more attention. And in the last couple of years, some interesting transgenic mouse models have been shown where class 1 restricted myelin-specific T-cell receptors have also been shown to be pathogenic in the animal model. So I think this is an area that definitely merits more attention, particularly as an effector cell, because their CD8 cells are much better candidates than CD4 cells.
Okay, so we have one final question from Genova. Are the anchor amino acids involved in TCR recognition a possible target for therapies aimed at modifying the pathogenic immune response?
Yeah, this is also an interesting question. I think we already have a therapy that is marketed that is based on this concept, even though the concept came after the drug was already moving to the market, which is copolymer 1. This is a randomized peptide mixture that has certain amino acids, alanine, tyrosine, lysine, and glutamic acid in a certain molar ratio. And this randomized mix of peptides that binds to MHC and has at least some of the preferred anchor amino acids actually works as a therapy. It's only moderately effective, but very well tolerated, and is already used since more than 10 years. So in principle, this knowledge of anchor amino acids and peptide mixtures that have certain characteristics and silence or quiet down the immune system is something that is already in use and probably could be improved.
So actually, there's many questions coming in. Just a couple of quick answers, maybe. So this is from Mauro Malnati. How much do you think the aberrant recognition of MBP is crucial in the pathogenesis?
The aberrant
recognition of MBP.
Yeah. I mean, as you saw, we used MBP primarily as a model organism. And I think that the entire field that has focused on myelin, because myelin is destroyed in MS, this may very well be wrong or not as important as we think and claim. Because there are autoimmune diseases like primary biliary cirrhosis, for example, where the autoimmune response is against the mitochondrial energy metabolizing enzyme that is ubiquitous in our body, but the autoimmune response is exquisitely specific for small gall ducts. So to translate this to the brain, there could be common autoantigens that are everywhere in the body that are important in MS, and our focus on myelin proteins could be, I don't want to say the wrong way, they are certainly not unimportant, but less important than we think right now.
Okay, the very last question comes from Chieti. To your knowledge, which of the new monoclonal antibodies is the most promising in the treatment of MS and why? Or do you believe other strategies are to be preferred?
I will give you a totally biased answer. I think that the anti-CD25 antibody daclizumab is the most important. The reason is that different from others that deplete certain immune cells for long periods of time and therefore carry important risks, like antibodies that deplete B cells or B cells and T cells and even monocytes, that this antibody enhances immune recognition or an immune cell population that is important, natural killer cells. It expands CD56 positive natural killer cells that are immunoregulatory and are also important for protecting us against viral infections and mutated cells. And that's why I think daclizumab is more promising. It may not be as efficacious as some other monoclonals, but I think in its overall profile, I think it's the most promising. But that is totally biased because we started to look at this first.